So today's lab, we're going to talk about the compound microscope, and then we'll look at oil immersion, and then we're going to look at the clinically important fungi, some of them, not all of them, there's a bunch of them. But I want you to know that everything we're talking about in lab is going to be on the practical, right? So need to find an effective mechanism to make sure that you are studying them, okay? And that you understand them. So let's go ahead and get started. So first of all, I want you to know that this is called a compound microscope because we use more than one lens, right? And so the lenses are here. They're also called the objectives, right? So these are them right here. And the other lens is the eyepiece of the ocular. So if we look at our microscopes that we'll use are going to have 4 objectives. One will be the 4X, right? It'll have a red line around it and a 4 on it. That's the scanning lens. One of them will be labeled 10X. It'll have a yellow line around it, and it's the high power. One of them have a 40X on it, have a blue line around it. That's the high dry. And that one is used to look at fungi and algae and things like that, right? The 10X is used mostly for looking at larger structures like plants. Could be, but you really could be smaller structures, but you really need the 40X. And the 100X is used to look at bacteria, and the 100X is called the oil immersion lens. Okay. So if you wanted to know the total magnification for the field of view you're looking at, you would multiply the eyepiece, which has a magnification power of 10 times, right, by um, the objective, which are going to have different, right? So a 4X and the eyepiece will have a 40X total magnification. 10X will have 100X, 400X for a 40X, and 1,000 for the 100X. Okay. So we call the 100X objective the oil immersion because it's so powerful that in order to be able to see the structure correctly, we need to have something that's going to correct for that refraction index, and we'll talk about that in a minute. Okay, so I want you to know these parts of the scope, right? Now, when we get to working with the scope, when we go back on campus, we will have a review practice with the microscopes. Because if you've never used an oil immersion lens, it's going to be challenging at first, right? But I want you to be sure that you know the following parts of the scopes and what their purpose are, right? So the first one, of course, is the ocular or the eyepiece. It has two functions. Number one, it's a lens, right, in a compound microscope, but also, secondly, it brings the field of view to your eyes. Okay. The body of the scope is this entire structure. If you come through here all the way down, and that supports all the other parts of the scope, right? This is the revolving nose piece, and its mechanism is that you can um, rotate those objectives around this nose piece, right? And all of these objectives are said to be parafocal. Has anybody ever heard of the term parafocal? Okay. What does that mean, Miss Amandetta? What does that mean?
That like you can use one lens and get it in focus, and then you can switch to another lens and it should still be in focus. You only have to fine tune it a little bit.
Perfect. So they're all aligned with each other, okay? And that's really important because our scopes, when you use certain scopes, you're looking at one and you want to go to the next, and you have to start all over. You don't have to do that here. You can just move from one to another, okay, including oil lens, okay? I want you to know the objectives, right? And so there are four of them, and I talked about them already. We'll talk about them a little bit and a little bit also, right? But they are really the scanning, which is at 4x, the high, which is at 10x, the high dry, which is at 40x, and then the oil immersion lens, which is at 100x. Okay? And if you multiply those by 10 for the eyepiece, then you get the total magnification for the field of view that you're looking at. So this would be 40x, 40 times. This would be 100 times. This would be 400 times. And this would be 1,000 times. Okay. I want you to know what the stage is. And, the stage itself gives rise to the condenser. This is the condenser down here, right? So be sure you know that. It condenses light from the lamp source, right? And then you have a light intensity knob right here. There is a little lever, and that little lever is called a diaphragm. A lot of people might call it the iris diaphragm. It's just a diaphragm, okay? And the diaphragm, along with the condenser, will allow you to regulate the amount of light that's coming into the microscope. And so the rule is the higher the magnification, the more light you're going to need. Okay, so you're going to need a lot more light when you use the oil immersion than if you were using, say, the scanning lens. Okay, good. The other things that I want you to know about is there's going to be some stage adjustments. Usually there are two knobs. One of them will allow you to move the stage from left to right, and then the other knob will allow you to move it upwards and backwards. Okay, and so that allows you, once you get your slide focus, you can just move the stage and the slide will move with it. Okay, and then the course adjustment is, its function is to bring things into as close to focus as possible. Some people can just use that one and that's all they need. The other one is the fine adjustment knob, and this is the one that you're going to use to look at fine detail. Okay, so I have um, done a similar thing, but this is me talking in more detail about the microscope. And so this is a video. It's embedded in this particular presentation. So if you want to look at it, you can try to look at it in the presentation. If not, because some computers won't let you do that, I've placed the link to the video here, and so you can copy that link, put it into your browser, and you will find um, that video on YouTube. Okay, and you can watch it. There are some differences in microscopes quality throughout the college. This is one that you'll see at HLC right here. But this is one that you'd see at Riverside. And they both work equally well. And so you should have no problem using the scope. Okay, so be sure you know the different. This might be reviewed for a lot of people, but be sure that you're very, very familiar with the different parts of the scope. Okay, any questions? All right. So when we're using the 100x objective, the oil immersion lens, the lens is so powerful that if you try to look at the images that you want to look at without having any kind of correction, there's going to be a lot of distortion. And so what we use for correction is immersion oil, right? So the way this works is that because remember, Amendetis already told us that all these lenses are parafocal, so they're all aligned with each other. So if you want to go from a high power to the oil, all you got to do is move the lenses just a little bit, place a drop of immersion oil on the actual slide itself, and then move the oil immersion lens, the 100x objective, so that it makes contact with the oil. It should make contact with the oil, but not with the microscope slide. Okay? So if you think about that for just a minute, think about you might be at the swimming pool and you drop something in the water in the pool, but you don't want to go get wet. So what you do is you grab that pole with that net at the end of it and you try to fish it out of there. When you put that pole into the water, if you look at where that pole enters the water and then you look at the pole in the water, there's going to be a little bit of distortion, so it looks like the pole is a little bit skewed upwards or downwards, right? That's um, kind of a refraction problem. And so that's the same kind of problem we're going to have here with this microscope lens, this 100x objective, this all immersion lens. And so we correct for that refraction by adding a little bit of oil, and that corrects it. So you'll be able to see things clearly. Okay. So here's a video of me using the oil immersion so that you can see how it's used. Please watch this. We'll work with it when we get back to in person also, right? And then um, this is what you'll see if you were for this particular slide that I used. So you can see that I'm pointing to some different structures that um, have. I would like for you guys to be able to tell what these structures are, right? So the first thing is easy, and so you can see that the the red arrow is pointing to A. Can somebody tell me? Can somebody tell me the morphology of that particular bacterium? Can somebody tell me the morphology? What is the morphology?
Shaped,
Uh-huh.
right?
What is it? It's a
The
bacillus. That's correct.
rod-like.
It's a bacillus. It's a rod. That's correct. Very good. Now, the other two things are looking at artifact. Artifact are on the slide, but they have no bearing or relevance to what you're looking at. Sometimes artifact, like what's under B, right, are crystals, and sometimes these crystals form when the slide dries too fast, right? But those crystals have no bearing on what you're looking at. It's just artifact. The other one that's labeled B is a little bitty, probably a particle of dust, right? And so you can see that it has no real true shape. It's an artifact. So I want you to be sure that you understand when we use the word artifact, that artifact means that it doesn't have any importance to what we're trying to study. Okay? The one that's the red arrow, that is important. That's the bacterium itself, right? But other things that are important that you might not appreciate just yet is that there's some inclusions here. There's an inclusion there, there's an inclusion there. So there's some inclusions in these bacteria. Some of them might be spores, it could be other things, right? And so we're going to be looking at some of those inclusions as we get further on into the course. So I've got the um, link to the video. If you can't look at it embedded, you can just copy that and then you can watch the video, okay, into your browser. Any questions? All right, so now for the main event. Okay, so if you want to define this particular presentation, you go on Blackboard, you click on Labs, then you click on Practical 1, and then on Practical 1, you'll see that there is a folder for this particular presentation. There's some other things here also, right? So there's a a study aid, there's a matrix. A matrix is really important, and then there's a review. We're going to be doing the review together in 2 weeks, but these other things are really important. Now, let me show you what they look like. Right. So the study aid is a list of all the organisms you're going to be responsible for. Right. And so there's a list of them. You can acquire that through that particular um, um, file on Blackboard. The matrix is um, a, it is something that I've created that has all the organisms you're going to be responsible for, and it tells you all the characteristics and what it's important for those about those characteristics. So these are for you all to allow you to get ready to take the first practical. And the first practical will be the week that we go back to face, and it'll be on that Thursday. Okay, so that's going to be the 4th of February. So it's already coming up. So don't let this, you should be studying this stuff a little bit. All the stuff we're talking about is going to be on that practical. So as I see it, today we're doing the fungi. On Tuesday, we'll do the protozoans. And next Thursday, we will study the metazoans and the exoparasites. And then we'll go back to class, and we'll have a review on that Tuesday. And then on that following Thursday, the 4th of February, you'll have your first practical, right? Which then will be followed up pretty quickly thereafter by your first lecture exam. So you can see we're in the first week, but there's already plans for the near future. Okay? Does anybody have any questions? All right.
Yeah,
It's
I can't
under.
find the oil one either.
I'll show you in a minute. I'll show you in a minute. Okay? I'll show you in a minute. Um, okay. Let's go ahead and talk about the fungi. So the fungi themselves, um, we there are two types of fungi that we're going to be interested in. We're going to be interested in the yeast and the molds. So if we're looking at yeast, we're mostly going to be looking at whether or not they bud. Now there's some other things that the yeast do that I'll be able to show you so that you can use them diagnostically. But the molds are a little bit more complicated because we use two different things to classify them and to identify them. Most important is the spores when we're looking from a clinical perspective. The second thing is the mycelia, right? The mycelia is a vegetative structure, right? We talked about them when we were talking about our lecture and we were looking at the fungi, and I said, hey, we classify them with spores, but also it's the vegetative structures that put them into a group. Okay, so I'm gonna make sure you understand those things today. So there are lots of books and lots of texts that have been published about helping people identify fungi. Uh, I have a book that's just on yeast, and it is about the size of uh, the biggest book you can think of, right? It's huge. Um, but that's just yeast. There's all these other things that are molds, right? And so we're going to be looking at those particular structures in just a minute. So these are the organisms you're going to be responsible for. You're going to be responsible for Candida albicans, right? It's a opportunity. All these are opportunistic pathogen, except coccidioides, which is a true pathogen. So you're going to be responsible for Candida albicans, and I'll show you how to identify it in a minute. You're going to be responsible for Saccharomyces cerevisiae, right? It's an important organism, um, but only as a yeast, because all yeasts look alike, except with Candida. So I'm just going to expect you to know that that is a yeast. Candida albicans has some special structures it has that will help you identify it, so it's going to be easy to do so. The other organisms are the zygomycota and the ascomycota. These organisms are less evolved, right? So we just simply call them the lower molds, if you will. And the ascomycota are higher. They're more evolved. And the evolution of these things is going to be based on their mycelia. Excuse me just 1 minute. Okay, so mycelia are the vegetative structures. They're the body of the mold, right? The spores are the reproductive structures, right? And so the mycelia can have two different forms, and they're going to look like this: long strands, and they have nuclei in them. These are called aseptate because they don't have any separations. Okay. The other type is associated with the higher organisms, and the mycelia is going to look like this. It's going to have separations, cross walls, and it's going to show you the individual nucleated cells. Those are called septate mycelia because they have separations. Okay? Any questions so far?
So the aseptate are those with the lower, the zygomys,
That's correct.
okay.
That's correct. Okay? Let me show you what I mean. So here is a mold growing on an agar plate. And here is mold growing on somebody's bread. Okay, my bread. But the fuzziness are the mycelia. Okay? Can you all see these little black structures here? All these little black structures? Those are the spores. Okay? Those are the spores. Now you can't see them as readily here, but they're there. You can see them really well here. Right? This is an organism called Rhizopus, and we're going to talk about it in a minute. It's a fast grower. So for instance, if you look at your bread on, I don't know, Monday, and there's a little bit of mold growing, if it's Rhizopus, you come back Tuesday morning and your bread is completely covered because it grows that fast. Okay? So that mycelia can either be aseptate, no cross walls, or it could be septate, has cross walls. Right? And so if we look at this for just a minute, here you can appreciate the difference, right? So here you can see that they have cross walls, right? This is septate mice. It's called a hyphy. Now that's just an idiosyncratic thing that happens to some people who talk about these things. They call mycelia hyphy, but hyphy is an individual cell. So one of these cells is hyphy, but the entire thing is mycelia. Okay? So this is septate, but this one is aseptate. There's no cross walls. Okay? And then it gets a little bit more interesting when you have certain organisms that are yeast. And you know they're yeast because they have budding cells, but they produce these elongated cells that are called pseudohyphae, and pseudohyphae are essentially false structures, right? And so a lot of people get them confused if they don't know what they're looking at, right? So the key is that you want to be looking for um, yeast, right? So again, if I was to draw, if I was to draw septate mycelia, they would look like this. There's cross walls, and then each of those cross walls have a nucleated part. Okay. The ones that are labeled aseptate, right? Um, they have the mycelia, but then they have a nuclei, but the nuclei have no separations. Okay. And the ones that are pseudohyphae are elongated yeast, right? So now you have elongated yeast. And on those, you will have budding yeast, and that's the giveaway. So it appears that maybe I updated this just recently and I didn't put the updated version on Blackboard. I will do that right after we get out of class today. Okay. So everybody see the difference? Anybody have any questions? You
I
will also have you will also have this video. It's recorded, so you can go back to it and use it as a resource.
I
Yes, Ava.
see the difference, but I got a little lost about like where we started. Like why are we talking about? Like what's the big
Why
picture
are we
of this?
why are we talking about this?
Yeah, like what does it tie
Be
into?
oh, good, because this is how we identify and differentiate the fungi.
Okay, got it. Thank you.
Okay. It's gonna make more sense in just a minute.
Okay.
Okay. All right. Now, here is a slide I put in to talk about the difference between the mycelia and the hyphae. So the mycelia are an aggregate of cells, right? They're multinucleated. But what is a hypha, or what is hyphae? Somebody tell me, what is hyphae?
Isn't it like the cells in the mycelia? Just
Uh-huh.
a single cell?
Exactly. It is a single cell in the mycelia. This is a hyphae. Okay? Anybody have any questions? So now, Ava, I'm going to put it all together, right? So this is yeast. How do you know it's yeast? Tell me. How do you know it's yeast? It's budding, right? So this, excuse me, this is a unstained slide, right? Right here, unstained. And so you can see it's kind of hard to see. So we can stain the organisms and it makes it easier to see. The problem is once you stain something, you kill it. So these are alive, these are dead. Okay, you with me? So for Saccharomyces cerevisiae, I'm going to show you an example of this and I'm going to ask what kind of organism it is. And if you say yeast, it'll be correct. But if you get more technical and say it's Saccharomyces, it'll be correct also. Okay? Because there's no really good way to morphologically distinguish this particular cell or this particular slides with a specific yeast because they all look alike. Okay? Only if the yeast have other structures that they produce can we make some determination about them clinically. Okay? And the one organism that does that is Candida. Does anybody have any questions about yeast? Okay, this is Candida, right? So I've used color coded my arrows to help you see things, right? So the top, this is Candida albicans, right? It causes thrush, oral and anal thrush. It also causes yeast infections, vaginitis. Okay, so if you look at the top slide, the top slide is a slide from the vagina of some female. And so you can see the epithelial cells in the background, but you can also see the budding yeast at the golden arrow. Can everybody see that? Everybody with me? Okay. The other thing that I'm pointing at with the blue arrow is the pseudohyphae. So if you put those things together, if you see pseudohyphae and you see budding yeast, then it's going to be Candida albicans. Okay. Now the top image is perfect. You couldn't ask for a better image. A lot of times what we see is what we see in the bottom image, right? And the bottom image is, again, you see pseudohyphae at the blue arrow, can you see budding yeast? Do you see budding yeast in that lower image? They're right there. See them? Uh-huh. A little. Yeah, yeah. So I wouldn't show you something like the bottom image. I would show you something like the top image. But I want you to know that, being a clinician and being a nurse is not an easy job, right? You're going to have to, a lot of times you're going to have to know you're going to have to take your knowledge and apply it, right? This is why you take a course like this, so that you can have the knowledge and then use that to help your patients. Okay? Does everybody understand that if you see pseudohyphae and budding yeast, that the organism is Candida albicans? Everybody good? Okay, very good. I'm going to show you now what it looks like when you have an infection by Candida albicans. There it is. So here you can see this is oral thrush. The top image is oral thrush. This is growing on your tongue right here. And then this is growing on the soft palate. You can see the tonsils and the uvula back here. But here you can see the yeast growing on the um, soft palate of the mouth. Sometimes they grow on the sides of the cheeks on the inside, but usually it's going to be either tongue or soft palate. Okay, and then the bottom image, what you're looking at is inside a vagina. You can see the cervix, and then you can see the overgrowth of the yeast in the vagina. Okay, good. So how do you acquire this? Well, Candida albicans is part of the um, normal inhabitants of your mouth and the vagina and the anus. They're there all the time. There needs to be some kind of disruption of the normal inhabitants there, so that, yeah, so Sarah, that would do it. So if you take an antibiotic, it's going to kill all of the bacteria that are susceptible to it. And now the yeast have all this space to come back to. So that's typically what we would see with um, vaginitis, right? This is due to the antibiotics you might be taking. This one up here, not so much. This one, when you have oral yeast, a lot of times you're stressed out. Um, so when you get stressed out, you will likely be more susceptible to infections of the respiratory tract and the GI tract. And so then you can develop this. Now, it can happen in kiddos that are practicing good hygiene, and in babies who are breastfeeding. Sometimes the breasts get infected with yeast, and the babies can acquire that and then get oral thrush also, right? But we do such a good job nowadays with communicating to mamas about how things can happen, and so it's not very common. It still happens, but it's just not very common. Okay. So I'll give you my personal story, because I think personal stories are important. Um, Several years ago, I was working at Johnson and Johnson, about 60 hours a week. I was teaching 2 classes at ACC, and I was working on my second master's degree in health services research. I wanted to be an epidemiologist. I was being trained to be an epidemiologist, so I was pretty stressed. And so one morning I woke up and my throat hurt, and so I looked back there and I saw all this white growth back on the soft palate, and I thought, oh, maybe I have strep. So I have my own microscope uh, here at the house. And so what I did was I took a little bit of a sample from the back of my throat and I put them on um, the microscope, and I saw yeast, and I said, oh, heck, I've got thrush. So I went ahead and stained. I have my own staining techniques, my own staining apparatus here. I went ahead and stained it, and I took it to my doc. So I was working in Round Rock, and the doctor was in Round Rock. I took it there, and he and I always had a good relationship. Um, And so I came in and he said, well, what is my favorite microbiologist here for today? I said, I have thrush. He looked at me and said, no, you don't. I said, yeah, I have thrush. He goes, and I said, here's a couple slides I made at the house. You can check it out. And so he gave it to one of his technicians and they went to put it on the microscope for him. But he didn't really have to look at that because he said, let me take a look. Once you look back there, there's nothing that looks like thrush. This is what it is, right? And he said, yeah, you have thrush. He said, well, what does my favorite microbiologist want as a treatment? I said, I'd like a 3-day treatment with diflucan. So diflucan is one of my favorite drugs. It's an antifungal. It's also generically known as fluconazole. And he said, oh, good choice. He said, you can also use nystatin. I said, nah, it's too long. I wanted to be treated and feeling better right quickly. And so I took my first diflucan and I felt better that first day. Right. Diflucan is an outstanding drug, right? An antifungal. Any questions about how these things interact? It says, well, you got to have a relationship with them, okay? Yeah. So nowadays, physicians are getting a lot better about it. I went into one place with one of my friends. He felt bad, and he said, "What can you do for me?" I said, "Nothing." I said, "You have strep throat. I can't do anything for you." I said, "We need to go and get you some antibiotics." So we went into one of these little places where you can go and be seen. I told him we should go to CVS because it's cheaper, but he wanted to go to this place because he knew somebody or whatever. But I was impressed because on the inside when you walked in, they had this big old poster that said, "We do not prescribe antibiotics for, or we only prescribe antibiotics for bacterial infections," right? Which I thought was pretty great because a lot of people don't know the difference between bacteria and fungi or yeast, and so it's important that we be sure that we educate them about the difference. That's why you take a course like this. Okay. Any questions about the yeast or about Candida albicans specifically? Okay. So now let's move over to the lower fungi. There's only one example, and that's Rhizopus stolonifer. And what I want to tell you about this is, remember I told you it's about the mycelia. There are no cross walls in this mycelia, right? So it's a lower fungi. Well, the other thing I will tell you about the lower fungi is that their spores are inside a vesicle, right? So if I was to draw the vesicle, the spores would be inside it. So although I don't claim to be a really good artist, I'm drawing this part right here, okay? And this part right here. The spores are inside, okay? What the giveaway is for this particular organism is at the red arrow. Those things that look like roots are not roots. They are rhizoids. And rhizoids for this organism are used for attachment, right? So usually roots are conductive. That means that water and nutrients will go through them. These are not conductive. They're just simply used to attach to whatever these organisms are growing on, okay? Now, while this organism is opportunistic, you have to be immunocompromised to get an infection by Rhizopus, but it's only going to grow. So the mold will only grow where there's a lot of oxygen because they're aerobic, right? But the yeast can be what we call facultatively anaerobic. That means that they prefer oxygen, but they'll grow without it, right? And therefore they could grow anaerobically. But the molds will only grow with oxygen. So they're only going to infect the parts of the body that have a lot of oxygen. Can you name those parts of the body where there's a lot of oxygen? Lungs, good, like it, Cesar. What else? Not the blood, not the blood, although I guess there could be some there, but not usually. So that's too vascular. So what about the surface of the body, the skin? is the other big thing. So typically we're going to see these fungal infections in the lungs and on the skin. Okay? Questions? Okay. So let's go to, we're almost done. Let's go to the higher fungi, right? And so we're going to talk about 4 organisms, right? Penicillium, Aspergillus, Microsporum, and um, Coccidioides. Now, all the organisms we've talked to, that we have talked about or are going to talk about today are opportunistic pathogens. Okay, the only one that is a true pathogen is Coccidioides immitis, and we're going to talk about it at the end because it's really important. Okay, so if we look at Penicillium, right? So the structures, of course, are easy to distinguish because they look like a broom or a brush or a hand, right? And so if we think about it, the mycelia comes up like this, and these are higher fungi, so they have septations, right? They come up like this, and then they give rise to these other structures that are called, I'm not going to draw them all, but these other structures are called phyalides, right? And then these phyalides give rise to spores. So can you all, if you look at this structure right here, see those structures that I tried to draw, although it doesn't look very good because I'm not a very good drawer. Okay, well, let's practice. Okay, so let's practice. What is this thing right here? What is this thing right here? Somebody help me out. Not phyalide, not the head. No, this structure, the septate mycela. Very good. That's what it is, right? Okay, so if I extend this out a little bit and then point right here, what are these things? What are these things? Oh, you're right, Sarah. It is a conidium. How did you know that? You're right, the head, yeah. But those are phyalites, yeah. So the entire structure, oh, it's not. The entire structure is a conidium, right? But those things that I'm pointing to with number 2 are um, the phyalites. Number 3 would be the conidium, the entire thing. And then what are these little round structures here? What are those? Spores. Very good. So now you know, right? And so if we think about that for just a minute, um, here you can see is one that is not stained, right? And they're harder to see, right? And so we stain them to make them easier to see. Okay, I'll make sure that I use a really good image when we do the when we have a practical. All right, let's talk about. Anybody have any questions about penicillium? Anybody have any questions about penicillium? All right. Let's talk about Aspergillus. So Aspergillus, remember I said that the higher fungi, their spores grow outside of a vesicle. Now with Penicillium that has no vesicle, it goes right from mycelia to phyalide to spore. But if I was to draw, and I'm not very good, but I'll do my best. If I was to draw this organism, so here would come, this is the vesicle. Here's the mycelia. Then from the vesicle, you have the phyalides. And then from the phyalides, you have the spores. So can everybody appreciate what I'm trying to show you on both of these images? Now, the image on the left was done by Eileen Rojas, and I left her name on there because it's so beautiful. It's so easy to look at it, right? And so can you see where the vesicle is and how the phyalides are coming off and then how the spores are coming off, right? Can everybody see that? Right? Somebody say yes. Okay, good. Now the spores are called Candida. Sometimes they're referred to Candida spores. If you call them spores, Candida, or Candida spores, it's all correct. Okay. What about on the right? Can you see where the vesicle is? The vesicle is right here. And then off the vesicle are the phyalides. And then off the phyalides are the spores. So that's an easy way to tell, to distinguish um, Penicillium from Aspergillus. But the other giveaway is that Aspergillus is always going to have a foot cell. So on the red, with my red arrows, can you see the foot cells for both of these representations? Can everybody see that? Okay. So if you see foot cells, then it's going to be Aspergillus, at least for us right now. In real life, with all the organisms in the entire world, there are other organisms that have a foot cell, but for the ones that we're studying, Aspergillus is the only one that's going to have a foot cell. Okay? So if we compare the two organisms, because people get them confused all the time, can you see the difference morphologically between Penicillium and Aspergillus? Can you see the difference? Okay. So if I was to draw a little quick draw, Mycelia, phyalides, looks like a hand or a brush, and then spores. Okay. If I was drawing Aspergillus, Mycelia, vesicle, phyalides, off of the vesicle. Remember, higher fungi, the spores are on the outside of the vesicle. And then you have your spores. Okay, questions? Oh, well, let's not forget about that, Miss Sarah. So I'll draw a little quick foot cell here. Yeah, yeah. I try to not draw as much as possible because I'm just not very good at it. Okay, foot cell. It's supposed to look like a foot. It's supposed to look like this. Okay. Questions. Now, why are these important? Well, my friend that I used to work with at J&J, her husband got cancer, and he was undergoing chemotherapeutic treatment, and he had been given the all clear sign. The cancer was in remission. He felt so good that he decided to mow the yard. And when he mowed the yard, a fungus got into his lungs. And because he had no immune system, because he was on chemotherapeutic drugs, the fungus killed him. The mold killed him. Isn't that just terrible? This is why we study these things, right? Any questions? All right. Two more, and then we'll call it a day. So this is the easiest one of them all. This is Microsporum Canis. It causes ringworm. There are three organisms that cause ringworm, right? And they are trichophyton, epidermophyton, and Microsporum. Now the other two, trichophyton and epidermophyton, are hard to spell and hard to say. So I picked Microsporum because it's easy, right? Some people might call it Microsporum, and I guess that's okay. Okay. So, but if you look at these organisms, they're so easy because There's only one Candida or one spore that's formed off of the Mycelia. Okay, so can everybody see that? There's only one spore, and that spore is huge. It looks like a big leaf. Are you with me? So these are what these organisms look like when they're growing, and then we put them under a microscope. We can tell, this is a ringworm, and we probably could tell just by looking at where it's growing on the body real quickly. And most ringwormers all treat it the same way. So I don't really have to do any type of speciation, although I will if they want me to. But I'll just call it, tinea, right? So it's interesting that the medical institution does not like to say Microsporum epidermophyton or trichophyton. They came up with their own word, right, called tinea. So if you have tinea, that means that you have ringworm, okay? Now, to make it even more scientific sounding, they decided to make the species name, that's the genus, the species name, on the place of the body where it was growing, right? So Tinea capitis would be ringworm of the head. Tinea corpus is ringworm of the body, right? And so this is what they look like, right? So you can see that baby's got ringworm. That's a pretty serious infection of ringworm. That poor baby's itching like crazy, right? Um, and then the lower image shows Tinea capitis microsporum of the head, right? And so a lot of times when you have ringworm, the cells that are associated with the infection, um, the hair follicles die, and so the hair falls out, right? They're not dead forever, but the hair does fall out. Okay. Any questions? So if we look at Microsporum, how would you describe the morphology of the mold for Microsporum? How would you describe the morphology of the mold? A leaf, right? So you have mycelium. Oh, let me draw it. You have mycelia, and then it gives rise to a single spore that looks like a leaf. Okay? Questions? Um, okay, so help me understand that, Amy. So do you mean like if I have thrush, can I give it to you? Or let's just do it this way so I don't offend you. So if your husband has thrush, can he give it to you? Um, he can share it with you, but whether or not you come down with an infection of yeast, it's going to depend on his immune system. It would be easier, and a lot of times, again, I don't want to offend anybody, so I'm going to not use anybody as an example, but when you're having sex with somebody, sometimes the penis can have yeast on it and you just don't know, and when it gets inserted into the vagina, it can transfer that yeast into the vagina, and then the little tears that occur during sex to the vagina can allow for the yeast. If you are having some kind of issue, you're taking antibiotics or something, it can allow the yeast to be transmitted that way. But in general, Amy, I would say that yeast are not transmittable. I mean,
Okay.
it's just
So like
okay.
ringworm in the baby, that's just
Oh,
his.
ringworm is not a yeast; it's a mold.
Okay, okay. Oh, yeah.
That one is transmittable.
Okay.
Okay. So yeah. So babies can spread it to each other, and kids can spread it to each other. You know, Amy, if you had to have an infection, ringworm is the one to get because it's easily treated. All right. It just looks bad. Okay. And you can get it from your dog too. If you look at that genus and species, Microsporum is the genus, Canis is the species, and Canis means what? Canine, dog. Yep. Good. Any questions? So Amy, I hope I answered your question without confusing you. Okay, last one for the day. Uh, let's talk about the bad boy of the group. So this is Coccidioides immitis. It is a true pathogen. And so there are many ways to identify this. We could look at the spores. We could look, there's just it has two different phases, right? We'll talk more about the different phases in lecture. But for here, I'm just going to provide you an easy way to identify it. So the mycelia is what you're looking at, right? So just imagine that um, this mycelia, and what you're looking at is the individual cells that have disarticulated from the mycelia. What that means is that the individual cells have broken apart, and each of those individual cells become what we call an arthrospore. And each of those particular individual parts of the mycelia can go off and cause infection or start a new colony somewhere. Okay? So while there's a lot of ways to identify this, this is the only thing I'm going to ask you about. This process of producing the arthrospores is called oidia. Okay? And it makes it real easy to identify this organism because it's one of the few organisms that does it, and we can use it diagnostically. So if I saw this, I'm like, oh my God, I've got oidia occurring. It's going to be one of the few organisms, and then if I have a little bit more background, I might be able to tell you what the organism is without doing any other work. Okay?
So
Good.
what's OID again? The process of it separate?
Oidia is the process of the separation of the individual cells in the mycelia. Okay? That's correct, Amenria. Each of the arthrospores are an individual hyphy. That's correct. Okay? Anybody else have any other questions? Okay. So now, this is a true pathogen. All right. It can cause problems in non-immunocompromised individuals, where most of the infectious agents that we talked about, we said there has to be something going on, right? That doesn't mean that it's not going to cause infection in the immunocompromised, but this one can infect other individuals too. And where did we say the molds are most likely to cause infection? The parts of the body that have most oxygen because they're aerobic, right? The term aerobic means that they need oxygen. Okay. What parts of the body, remind me, what parts of the body are likely to be infected by mold?
Lungs and skin.
Skin and lungs. Good. See how much you're learning? So here is a radiograph showing the infiltration of Coccidioides immitis into the lungs. And then the bottom image causes valley fever. And then the bottom image shows the organism growing on the skin. Okay?
So
Infecting
what
the
you're
skin.
saying that is if you eat a moldy bread, you won't get sick?
No, because if you're eating moldy bread, you're not eating this organism. You're eating probably Penicillium or something else. This one gets disseminated through the air. Okay? So check it out. Have any of you all ever seen those commercials on TV from Humera or Embro that basically says, be sure and tell your physician if you've been in certain parts of the United States where certain known fungal pathogens are known to exist. Have you seen that commercial? Yeah, this is what they're talking about. And so this organism is found in Southwest Texas through New Mexico, up through Arizona, and into the desert parts of California, right? And so sometimes when you're driving that area, you might see these big old signs, please keep your windows rolled up due to the presence of fungal pathogens on the soil. They're talking about this guy, okay? Yeah. My friend who grew up in Arizona, um, I got to meet her father, and she was telling a story about how she was really sick as a little girl, and her father said valley fever. And so immediately I got really interested, and so I paid a lot of attention to their story. But she said, I knew I was really sick because my father never slept with me, but for that whole month that I was sick, he never left my bed. He stayed with me the whole time. So what happened is they were in Arizona, and they were building a swimming pool in their backyard. And they were digging up, and so my friend breathed in the spores. She was a healthy young lady, a little girl, and she breathed in the spores and came down with a coccidioides infection, right? And so she was really, really sick for about a month. But then she overcame that. But she always tests positive for the antibodies to coccidioides limitis. So when I was working with her at this one of the last companies I worked for, anytime I needed a positive control for coccidioides immitis, I would just go take a little bit of her blood, right? And because it was always hot, it was always positive for coccidioides. Okay. Can you see the importance of these organisms? Right. I'm beginning to tell the story and help you understand why this course is important. Remember, the two things I told you that you should know every day you come to my course is, number one, where you stand on the course, and number two, why is what we're talking about important, right? So Ava, did I make the connection for you about why the mycelia was important as we were talking about these infectious agents, right? Yeah. So I'm always I always encourage you to ask those questions, because that's how you learn. Okay. Are there any other questions